The second lecture exam will be based on the objectives, which are available in Blackboard, and all of the points are reviewed in both the notes and the videos. So, on the viral life cycle, you'll be asked to put the various steps in order, such as attaching to a receptor on the outside of the cell, penetrating the cell, etc. You'll be asked to distinguish between the lytic and temperate viruses. Temperate viruses, they can have a dormant phase known as a prophage, where they insert themselves into the host chromosome and then can become lytic at a later point. We talked about the differences between prokaryotic and eukaryotic cells. Prokaryotic cells are typically smaller. They lack a nucleus and membrane-bound organelles. And there are two major groups of prokaryotic cells, the eubacteria and the archaea. They differ in their cell membranes, their cell walls, and to some degree their lifestyles. And so, for example, archaea, they possess branched lipids in their cell membranes. Sometimes these membranes form monolayers. Instead of peptidoglycan, the structure of the eubacterial cell wall, they have a cell wall made of pseudomurine. They are often found in extreme environments. Methanogens are all archaea. And in contrast, eubacteria compose most bacteria, and certainly most bacteria which we encounter in the environment and which can cause disease. Now, bacteria vary a great deal, and so therefore they vary in size, with some being smaller even than large viruses, while some are so large that they are a bit larger than the smallest eukaryotic cells. They can vary in structure, whether this be, their shape of being a rod, a round coccus, or a spiral shape. Some are colonial, and some can even have some degree of internal membrane structures, like a folding of the cell membrane, or even a membrane around the chromosome. Autotrophs are organisms which use carbon dioxide as their source of carbon, while heterotrophs must obtain their carbon from the biomolecules of other living things, like sugars. Phototrophs get their energy from sunlight. Lithotrophs oxidize minerals, while chemotrophs need to get their energy from the chemicals of other living things, such as biomolecules. Some of these chemoheterotrophs may be parasites, decomposers, or predators, and so this is a common bacterial lifestyle which can be varied. Cyanobacteria, in contrast, are photoautotrophs, and they release oxygen as a waste product. This was very important not only today, but since they predate plants by 1.5 billion years, they were the ones who introduced large quantities of oxygen into the atmosphere in the first place. Only archaea can be methanogens, which produce methane as a waste, and they can be found whether it be in swamp water or in the intestines of mammals. This methane can worsen climate change, so it is of interest. Bacteria can have different lifestyles, so some are aerobes if they require oxygen, or anaerobes if they cannot live with oxygen, or facultative aerobes if they don't require oxygen but can nevertheless survive its presence. There are other lifestyles, such as barophiles prefer high pressures, or thermophiles prefer high temperatures, etc. Nitrogen fixation is the incorporation of atmospheric nitrogen into molecules such as ammonia, and this is important because plants and animals take their nitrogen molecules from those which bacteria have fixed, and so therefore this is important for the nutrients which cycle through food chains. Many of the extremophile groups have specific traits which allow them to live in these harsh environments. And so an acidophile may have a thin membrane around the cell which protects it from acid. The cryophiles, which can live in very cold temperatures, have membranes which remain fluid, modified membrane lipids. Thermophiles have modified enzymes which do not denature at high temperatures. Okay, and so these can all be points raised on the second exam.